Okay, so good afternoon. I have to say that it's a great pleasure to introduce our speaker today, Dr. Rolf Marschalek. And Rolf is the director of the Institute of Pharmaceutical Biology at Goethe University in Frankfurt. And Rolf is a worldwide recognized scientist and an expert in the field of MLL rearranged acute leukemias. And actually, he's very well known, and he has made fundamental and crucial contributions on the molecular mechanisms that are underlying the pathogenesis of leukemias that harbor MLL fusion proteins. Also, I would like to mention that he's visiting us because this represents also somehow the kickoff meeting of our recently granted project from the Josep Carreras German Foundation. And also he's really actively collaborating with the group of Pablo Menendez. So this is the title of the talk. I'm sure you will enjoy a lot. Thank you.
So thank you very much, Maribel, for this kind introduction. I also welcome you to this seminar today, which is entitled, as you can see, about the insights in disease and pathology of MLL rearranged leukemias. So I have to turn this on. So when you are talking about cancer and when you're thinking about cancer, then completely different concepts have been pushed forward over the last decades. For example, if you think about solid tumors, I know that you're mostly working here on liquid tumors, but just to keep in mind that solid tumors have an important factor, which is time, because with time and cell doublings, you increase mutations in cells. And what people did really nearly 20 years ago by sequencing genomes of colon and breast cancer, they found out that these cancer types are accumulating a lot of mutations. So they found up to 70 to 80 mutations in these cancers. When you go into hematomalignant tumors and think about hematomalignant tumors, it's completely different. Because here you need chromosomal translocations. Chromosomal translocations are shown here in this movie. You see two different chromosomes are recombining. And this is the only genetic event what you can mostly see in leukemias. So they can occur spontaneously, they can occur already in utero for infant leukemias. And so chromosomal translocations are the most important impacting factor for this. And what is also important, in many of these tumors, you don't see secondary mutations. So the way how these tumors are set on and these tumors are set on are completely different. And if you look really to the Sanger Cancer Census database and look to all the cancer genes which are known today, so there are more than 700, you can imagine that nearly 40% or more than 40% of the genes which are identified in hematomalignant, but also in solid tumors, are genes which are involved in chromosomal translocations. So let's also have a look to the prevalence of tumors and leukemias. I took only the data from Germany, but you compare this really one-to-one to Spain if you want. So we have in Germany more than 80 million inhabitants. We have nearly 800,000 newborns per year. We have nearly 1 million of death cases. And you see most of the death cases are coming from corona, heart diseases, but also, let's say, 1/4 is cancer. And we have every year in Germany roughly 500,000 cancer diagnoses, which is 0.56% of the population. So if you multiply this with your lifetime, you see that your personal risk is roughly below 50% to get cancer during your lifetime. And what is also important, we have half of them are dying every year. And this is when you look to Spain, you have a little bit less people in Spain, but you have exactly the same number for your personal risks every year to get cancer. So it's in Europe, I think it's always the same. And only 2% of all these cancer diagnoses are leukemias. And we have roughly in Germany nearly 10,000 cases of leukemia, and 1 in 1,000 newborns get leukemia already after birth. And of all these leukemia cases, 1 to 10% of these cases are MLL rearranged. That's the topic of my talk today. And we have now in the ML recombinome, so we have 118 different chromosomal translocations to the ML gene, which is causing acute leukemias. And as you maybe know better than me, because I'm not a physician, we can cure from these ML rearranged leukemia cases roughly between 25 to 40%. And the rest of these people are dying, and we need to work on this because we need to change maybe therapy or make therapies better in the future to get more people cured from these diseases. Today, I will give you insights in 4 different topics, what we are working in my lab. So I will really start on a very basic topic, which I'm really curious on to work on, namely the question why we have these chromosomal translocations at all. So what is the cause, the molecular cause for chromosomal translocations? Then I will shortly give you an overview of what our diagnostic center is doing. So I present you the 118 fusions to the ML gene, and then I will focus on particular ML fusions, namely an MLA4 and MLA6. And at the end of the talk, I will give you also some insights into a new project in my lab where we try to interfere with minimal residual disease as a major cause of death, because nearly half of the leukemia patients with ML rearrangements are dying of the relapse and not from the primary disease. So let's start with this basic fundamental scientific question, why we get chromosomal translocation at all. And I will start with something which most people are not really working on, namely with an interface cell nucleus, what you can see here. And you can see here that the decondensed chromosomes in different colors. So what you can see here is colored blobs are our chromosomes, which are tightly packed into this nucleus. And we call these areas of these chromosomes chromosome territories. And what is important to understand is how this is really looking when you look into detail into an interface nucleus. You see the tightly packed chromosome, and you see all these chromatin loops coming out from the chromosomal areas. And what is important is that transcription is starting in between the territories. And these places where transcription of genes is taking place, we call transcription factories. We have roughly 400 per cell. And if you think how many genes are really transcribed in a normal differentiated cell, which is roughly between 2,000 and 5,000, so in every of these territories, there are 5 to 10 genes transcribed together. But they are transcribed together also from different chromosomes. And so what is really important to understand, and this is also a very old story which has been published at the end of the 90s, when you really think what is going on, for example, in the blood cell, people have already shown that, for example, the MYC gene and the heavy chain of the IgH gene are transcribed together in such a factory, or B-cell and A-cell are transcribed together, or even the MLL gene and A4 are transcribed together. So there are many publications which have shown this, and this co-transcription is really taking place here. What is also important to understand, some of our genes have a very peculiar feature, which is also not very known. And this is that these genes are not making full-length transcripts, but they are making transcripts which are prematurely terminated. We call this early terminated transcripts, or ETTs. And we have already published in 2011, 2012, that many genes which are involved in chromosomal translocations do show exactly this phenotype, that they are not making full mature transcripts. They display in certain introns prematurely stopped transcripts. And this is true for MLL, but also for A4, AF9, ENL, also for the Ewing sarcoma gene, or for NUC98 or RUNX1. So we have identified in all of these genes these premature transcripts. And we have also control genes which are not making this kind of stuff. So why is this important? If you have a normal gene with exons and introns, we have splice sites at the end or in the beginning of exons and introns, of the exons, and we get splicing, as you know this from textbooks. But what is happening to a transcript which has this early termination? So you have a splice donor site which has no partner, no splice acceptor site. And what may happen are three different scenarios. One scenario is that in the transcribed intronic sequence, you find an artificial splice acceptor site, which is here. We call this a cryptic splicing. Or you take a second transcript from the same gene and splice into this same gene. So it is a so-called a cis or trans-splice event. So you get exon repetitions. So you get, for example, a transcript which has exon 1, 2, 3, 2, 3, 4, 5, 6, for example. But if genes are transcribed together, what can also happen is that you splice from this splice donor site into another transcript, which is co-transcribed in the same transcription factory, and you get fusion transcripts. So you get a fusion between two different genes by nature due to this fact that these transcripts have an early termination. Why this is so important? We have done this. This is a study from 2011. When you take 10 blood donors, which give you normal blood, and you analyze for MLL fusions, you find in every of these normal patients which have no leukemia an MLL4 fusion transcript. If you look for the A4ML, the reciprocal fusion, you don't see anything, because you see only the MLL4 fusion because of trans-splice events, and not because there's a chromosomal translocation. And you see this also for other known fusions, which are known, for example, for lymphomas, like the NPM-ALK fusion, for example. We did also race studies, so we used also the ENL gene or ELL gene, so we find fusions to other genes, but for the MLL, there's only the A4 fusion partner. So MLA4 transcripts are by nature in everybody here sitting in this audience, in your blood cells. So the surprising finding that this non-genomically encoded fusion transcripts, this is the acronym for this, occurred due to trans-splice events only at the RNA level, and they need gene proximity inside of the interface nucleus. So you need to have two chromosomes so near to each other that these loops can be transcribed together to make these kind of fusions. But you need also this early termination transcripts in this case. And so the important question is, can we mimic exactly such a situation, and can we induce chromosomal translocations by knowing exactly this, what is written here? And actually we did this. So we used ML and A4, and we used cyclin D3 and ML2. And we did this in a cell, in a kidney cell, where these chromosomes are far away from each other. So in this cell, these two chromosomes are far from each other. So they are not in proximity, so there should nothing happen. And we did a trick. We used a defective Cas9 protein, two different ones, which are fused to a domain, which is binding abscisic acid. So when we are putting in a drug in cell culture, this abscisic acid, then we are bringing these chromosomes together. And this takes about 2 weeks to rearrange the nucleus in a way that you bring these chromosomes near to each other. And we can show this by 3C experiments. 3C experiments is just a chromosomal capture technique. They really can demonstrate after 14 days that now chromosome 11 and chromosome 4 are near to each other by giving this PCR fragment. And the same happened for cyclin D3 and ML2, as you can see here from this band here. So 14 days of this trick, of this technical trick, brings in the interface nucleus the chromosomes together. What is happening? What is the consequence? If you look for MLA4 fusions, you see nothing on day 0, but you see the band coming up after 14 days. So just bringing these two genes in proximity causes a transcript, which is trans-spliced between ML and A4. We don't see this in the control genes that we used here. And we see by sequencing that we are fusing ML exon 9 to ML exon 4, like we described it before. So taking this situation on day 14, we induced now by a normal CRISPR-Cas system genomic breaks in intron 11 and intron 3 of A4. And we did the same with 2 exons which are compatible for translocation, namely the intron 13 from ML2 and the intron 2 for cyclin D3. And then we look what is happening, and we see, in fact, here's a control. We see the translocation is occurring in case of MLL4, and we can sequence exactly the ML exon 11, exon 4 fusion now, and also exon 3, exon 12 on the genomic level, and this is RNA. But we don't see anything for the control. So even if we have brought the two genes together, without the early termination transcripts, which are present here, even if you use CRISPR-Cas, you don't get a translocation. You get only translocation in case that the RNA MLA4 is present. So, as I told you, the MLA fusion in RNA is formed by proximity, is a transplice fusion gene, and this causes exactly the fusion that we know before, which is this fusion here. It didn't occur for the control genes, also they're in proximity. And when we induce the chromosomal translocation, we get exactly the fusion transcripts that we need to know, and which are in different exons. So it's here exon 9, here's exon 11 fused. So it's independent from the transplicing event, we can induce a chromosomal translocation. And when we sequenced the genomic bands between the genomic breakpoints, we found exactly the hallmarks of all chromosomal translocation, which are the typical facts that you get tiny deletions, duplications, and mini direct repeats at the junction. So this chromosomal translocations were exactly repaired like we find it in patients when they had a chromosomal translocation. So the conclusion is that we need two different things for a chromosomal translocation. We need, of course, proximity of the genes in the interface of the nucleus, but we need also the feature of all early termination of transcripts. Otherwise, you don't get a chromosomal translocation when you induce a DNA damage situation. And the DNA damage situation is, of course, in that case, a CRISPR-Cas system. Yeah. And if you take control genes without these early termination transcripts, even if they are in proximity, even if they are transcribed together, these genes, even if you induce a DNA damage situation, are not making chromosomal translocations. And this is quite important to understand. So all the genes that we know, maybe also BCR and ABL and so on, they have this kind of features which makes them vulnerable for chromosomal translocations. So I just summarize this. So this is the interface nucleus. I just draw now the chromosomes like we know chromosomes, but we are in the interface. We have here 4 genes. We bring together by this first CRISPR-Cas system with the defective Cas9 these genes together and bring it in proximity. We see fusion RNAs. We don't see any fusion RNAs here because only these genes have the ability to make early terminated transcripts. And then if we induce a DNA damage situation by chromosomal breaks, we get here a chromosomal translocation and here not. This is basically the story how chromosomal translocations occur. Good. So the conclusion, only genes able to form these fusion transcripts can also cause chromosomal translocations. Good. The next is a very short one, which is people who know me also know that we are since nearly 20 years doing diagnostic for MLL rearrangements with many labs all over the world, from Australia to Canada, from South America to Russia. And what people are doing, they are sending us a little bit of genomic DNA isolated from MLL rearranged leukemias, which were identified by split signal FISH or whatsoever. And with this genomic DNA, we did for nearly 20 years a technique which is called long-distance inverse PCR to clone chromosomal breakpoints. These chromosomal breakpoints went back to the centers, to the clinicians who use this as a molecular marker to monitor therapy and to identify MRD in case when the therapy was not working. And we use this information to define the ML recombinant, and so far we have 118 ML fusions known to be associated with acute leukemias, either acute myeloid leukemia or acute lymphoid leukemia. We can use the same techniques, of course, also for other problems, but mostly we are working on this stuff. And so we have really accumulated a lot of knowledge about different fusion partners, which are all associated with leukemia development. And this is the situation right now. We have now the data for more than 3,400 patients, most of them identified by PCR. Nowadays, we are making targeted NGS to identify novel fusions. And as you can see here, we have, in most cases, we see that the distribution and frequency of fusion partners is quite equal, but we get more genes discovered by this novel technique. And this is a situation, what we see, you see the major fusion partners here on top, A4, this is AF9, this is ENL, AF10, AF6. These are the most frequent fusion partners. And you see that in all these leukemias, we get mostly A4 fusions, followed by AF9, ENL, ML10, and AF6. And this is for sure different in ALL and AML, because in AML you see more AF9 cases, and you see more A4 cases in ALL patients. And we can subdivide this into different categories: infants, pediatric patients, and so on. And this is now the full list of all the fusion partners that we know nowadays that can cause leukemia. But as I told you, most of them are only 5 genes which are important. What is maybe important for you, when you see that so many different genes can cause cancer, that you understand that all these genes that you can see here are coming from completely different compartments of our cell. So they are cytosolic proteins, they are nuclear proteins. What is important, they are all causing acute leukemia. And what is also important, that in most of these cases, the same fusion partner causes the same disease phenotype. And as I told you before, the most important ones are here, written here. And this brings me to the next point, namely to ask the question, what are really these different fusion proteins doing in our cells? And I will tell you a little bit about MLA4 and A4ML, as well as about T611 fusion proteins. I will start with the MLL protein. The MLL protein is a very important protein in our cell. It's more than 4,000 amino acids long. It's being cleaved at a cleavage site by a specific protease called TASPase1. And it's assembling into a complex where we have on the very end terminus 2 different proteins binding menin and LEDGIF1. We have here on the C-terminal portion a complex for the Z complex. We have 2 different histone acetylases, and maybe there are also some other proteins binding to this complex. And the machinery, what you can see here is written here. The amino terminus of ML is something which can recognize promoters and can bind to proteins sitting on promoters, while the C-terminal portion here, the major portion, is chromatin binding, reading and writing. And so these factors here, this complex here, they can just bind to basic transcription factors like SP1, AP1, and so on, and some or MYC or NF-kappa B factor or whatsoever. So these are quite ubiquitous in binding to promoter factors, while this domain here is binding to histone particles and can read histone particles and can write histone particles. And what this domain is doing, it's writing on chromatin the famous signature for histone 3 lysine 4 3-methyl, which is usually found on active promoters. What is also important is the PhD domain, which is this one here, which has the reading function for histones, has also a switch function. There's an isomerase binding to this domain. And depending on the switch by this isomerase, polycomplex is recruited to this area here. And there you have, let's say, chromatin activating functions here, and you have chromatin repressing functions here. And so we have a battle going on promoters. And actually, based on ML knockout data, we know that about 1/3 of all genes are becoming activated for gene transcription, and 2/3 are shut down for gene transcription. So it has also a repressive function. And this is quite important, so you should keep in mind that the ML protein, the wild-type ML protein, has activation and also inactivation function for gene transcription, depending on the switch that you can see on top. If you use HDAC inhibitors and you block this kind of function, you can see that really the gene signatures are coming up, so you get rid of this repression function. And what is maybe also important for the project with Maribel here in the room, that if you somehow interfere with polycomps, which are the natural counteracting protein complexes by inhibitors of polycomp group proteins, you may also enhance MLL functions. So these kind of drugs and these kind of drugs are doing exactly the same at the molecular level. And this is actually the project we got funded together by this German Josep Carreras Foundation. What is also important to understand is A4 is a major fusion partner of MLL. So this is a T411 translocation. That these two protein complexes are working together. MLL is responsible for activating the chromotor area for gene transcription. And then you get gene transcription, you get a transcriptional start. Polymerase 2 is walking for about 50 nucleotides, and then it's stopped by these inhibitory complexes here. And this is quite necessary because at this point you get the capping of mRNA. So you see this termination needs or is needed for capping mRNA transcripts. And in order to get now this polymerase to work back to work, you need this protein complex, which is called in the literature the super elongation complex. And inside of this complex, there's PTFB, there's DOT1L, there's BRD4, there's another histone modification enzyme like NSD1. And so we are overwriting this block, and then you get an elongation. And you need for this elongation PTFB because you need to phosphorylate serine 2 of this motif of the RNA polymerase in order to get the pole R to pole A to the elongating polymerase. And in this, when you look at this picture, you understand what is going on. You get certain chromatin signatures in the promoter area, but also in the transcribed body of a gene. And these signatures are shown here. These are the famous signatures for active genes. And these signatures are giving, in a given cell, in a given tissue, a thing what we call transcriptional memory, because a cell knows by these signatures that this gene has been transcribed before and it should be transcribed again. And therefore there's behind MLL and AF4, there's also something which we call transcriptional memory. So what happens in these 118 cases of MLL translocation? This is again the MLL protein as a scheme. And what is important that nearly all translocations occur here. And what is then going on? We are separating these really tightly controlled functions, fix it by fusing translocation partner genes to it, and we are separating now the functions. And this is actually what we see when we have a chromosomal translocation. These now separated MLL portions, the N-terminus is really a very heavy activator of gene transcription. So we see target genes which can be upregulated 1,000-fold by having an MLA4 fusion protein expressed in the cell. And the other part, the reciprocal fusion part, can now act without promoter on chromatin. So it can activate chromatin all around your genome. It's not fixed now on promoters, it can do it everywhere. And you will see later in the data that I will show you that this is actually the function of the reciprocal fusion protein, that it can activate in a random fashion chromatin in a very short time. And this leads to a dedifferentiation process and to a novel transcriptional memory system, which is now oncogenic. And so you need both of these fusion proteins in order to get a cancer cell. And I will show you now this on data, but I will just show you some experimental evidences and not only these nice pictures. So what we have found out when we were analyzing these fusion proteins. Everything started already in 2004. This is a very old paper, it's nearly 20 years old, where we simply did, to that time, DOX-inducible gene expression. And you see this was a TET-off system to that time. So in the presence of doxycycline, we have no expression, and minus dox means that we have expression. We used to that time mutated RAS gene as a control. And you see in this kind of experiments that we get three-dimensional growth only when we turn on RAS, but even when we overexpress A4. So to that time, we already know that A4 is a proto-oncogene, because if we overexpress it, we get a tumor phenotype. And we saw to that time the tumor phenotype only with the reciprocal A4ML and not with the direct one. So the direct one without this one can do not so much. But it was really clear to that time that A4 is a proto-oncogene by definition. And when we use the A4ML, we also realized a very specific feature of this fusion protein, that we extend the half-life of this protein dramatically when we fuse it to a MLL. A4 has, as you can see here, A4 is a very unstable protein. It's heavily degraded in cells. The same is true, of course, here in the fusion protein, but the MLL portion is stabilizing this protein dramatically with a very long half-life of nearly 4 days. And this has oncogenic properties. We did then a mouse model by using both of them, and we realized in this mouse model that only the A4ML or both together are producing leukemia. And we can retransplant this leukemia many times, and then the leukemia onset is very, very fast. But it took us nearly 6 months, 6 to 7 months in order to get leukemia. So it takes really in the mouse system quite a long while. This is also true for other experimental systems where people have tried this in the past. So it's in some cases it takes really months, but then you get a full-blown leukemia. The immunophenotype of this leukemia was mostly pro-B. We had, but also cases where we see a CLIP phenotype, which means a common lymphoid progenitor phenotype, because we get B and T signatures, and we had also some cases with mixed lineage leukemia, so we get myeloid and lymphoid markers on the surface. And then this paper came up, and Pablo sitting in the audience, we know both very well this time point when this paper came up, where people said when we modify the MLA4, we are also able only with MLA4 to get leukemia. And this paper appeared, and so the question is, what is right? Are these people right saying if we modify the MLA4, then we get leukemia alone with MLA4, or do we really need both? And actually we wanted to prove this challenge, and what we did is a very, I think, intelligent experiment to find out a little bit more about the function of these fusion proteins. What you can see here is an experiment where we constitutively express MLA4, and we only for 2 days induced A4ML. You can see this also by color here. You see all the green cells expressing MLA4, and we expressed A4ML only for 2 days, and then it was turned off. And we see this also on protein level. These are plots from showing that MLA4 and A4ML is expressed even for this short time here on day 3 after turning on for 2 days the A4ML. Let's have a look what is coming out if you look to the omic parts. So this is RNA-seq data, and we don't have to look on genes, but I just want to show you some numbers. When we constitutively express MLA4 in a cell, we get 50 highly upregulated genes and 42 downregulated genes. Not very much. If we turn on day 3, for 2 days, the reciprocal fusion, we get 12 times as much target genes upregulated. We don't see anything in the downregulation. We get only upregulation. And the signatures, you can see that exactly day 0 and day 12 are quite similar, and they separate completely from day 3 and day 28. So we looked also after 4 weeks. And we were keen for this drop here and the coming back of these high numbers back again after 4 weeks. And when we analyze this in detail, when we analyzed the gene signatures in detail, we found a very interesting thing. So these are the genes up and down regulated on day 0, only MLA4. So these are all the genes, not only the high upregulated like this one. These are all the genes. We see a completely different shape in the RNA expression level on day 3 when we have the reciprocal. And we can trace this green signature until 4 weeks. We see this also for the downregulated genes. But we see on day 12, on this day here, also genes which are newly coming up, and we see even genes newly coming up on day 28. So what you can see here is that we set on an evolutionary process by reshaping the chromatin, even for a short time, namely only for 2 days. So we stopped expression of this protein here. This process is going on and going on and going on, even when we shut down the fusion protein which has turned on the chromatin. And we see here on day 3 an gene expression for innate immune response. So we see myeloid markers coming up here. And we see, interestingly enough, after 4 weeks, a B cell signature coming up here. And we see a downregulation of T cell signatures here. So there's an evolutionary process induced by 2 days of expression of the A4ML. And when we looked here, we did another experiment where we induced for 48 hours only MLA4 and shut it off. And look what happened after 4 weeks. We did the same for the reciprocal, and you see in both cases that you are losing the signature that were turned on by 2 days expression of these fusion proteins. By mimicking exactly this situation here, that we have MLA4 constitutive and turning only for 2 days on the reciprocal one, we see that there's a strong maintenance of genes. So this means there's a big difference whether you have only one fusion protein or you have both together. This was the story with T411, and the other one was T611, which is the most worst leukemia you can get because your survival rate is in the range of 10%. And interestingly enough, we had, when we decided for this project, we had only in our database 95 cases, and you see most of the breaks are in the major breakpoint cluster area, and you normally get AML when you have this translocation. But we saw also later on here in this area also breakpoints, which is a new minor breakpoint area of MLL. So it's after exon 21. And we see when having breakpoints here, we get T-cell leukemia. And I was fascinated by the idea that we can switch between TLL and AML just by switching here the breakpoint cluster in the MLL gene. And so we decided to make a very complicated experiment because we designed the normal T611 fusion proteins, which are shown here, but we also cloned the extended MLL AF6, where we mimic the breakpoint in intron 21 and not in intron 9. And we did also the reciprocal one. And we established 6 cell lines. So we are testing each of these fusion proteins alone and in combination, and then we get this extended MLL and the shortened MLL here, and then also the combination of both. And here you can see again what came out from the RNA-seq data. We see with MLA6 alone, with a classical one, only very few genes which are turned on and turned down. We see that the reciprocal can do much more genes turn on and turn off. The combination of both gave again the explosion, what you can deregulate. We get nearly also here 12 times as much genes upregulated, and we see here for the first time a massive downregulation of genes. When we switch the breakpoint, which means we are switching a domain which is encoded between exon 11 and exon 20, which is a PhD domain. This is a domain which makes a switch also in the MLL. When we switch this PhD domain to the direct fusion here, then everything is happening with this fusion construct and nothing is happening here again. The combination of both is more or less the addition of what happened in both cell lines here again. So we see this. And interestingly enough, in this population where we have the expression of both fusion proteins, we see an AML signature coming up. And in this here, we see T-cell signatures coming up. So it's quite interesting that these two systems here are behaving completely differently in our experimental system. And we expressed all this is happening after 48 hours. So we did the experiment exactly after 2 days. So what I just want to mention here, the PhD domain, which is normally in the middle of the MLL protein, has a very important function because this is a domain which something has to do with this chromatin opening function and which is responsible for the evolutionary process which is then taking place. Okay, so I conclude this one before I come to the last one. So what we show that these fusion proteins are really cooperating with each other. And the PhD domain containing fusion protein, which is either A4ML or AF6ML, or in our model, the extended ML AF6, they have the chromatin opening function. And this is important to drive a de-differentiation process. It allows an evolutionary process, and it potentially drives these cells into the pre-leukemic or leukemic state. And the other proteins are the strong transcription factors. They are turning, and in this open chromatin now, they are turning on everything what they need, and they are trying out what is helpful and what is not helpful, and then you get this evolutionary process. And therefore, as an experiment consequence, and this is what many people are right now doing, is when you really want to study leukemias, use CRISPR-Cas, because this causes exactly that two fusion proteins are being made in the same cell. And don't try this complicated stuff that I'm doing here by testing every fusion protein alone and then both together to find out what people can see when they're nowadays using CRISPR-Cas technologies. Good. This brings me now to the last part, and this is maybe the most important one. As I told you before, and looking now to infant leukemia, in infant leukemia, 66 of the treated kids get a relapse, and 80% of these relapse patients die. So we have here a real problem, namely the major cause of death during treatment when we have leukemia. And I will just show you here some interesting data that we found out. What you see here is the typical stuff, what you are doing with having somebody with a high-risk patient, leukemia patient. We have a schedule for 2 years of chemotherapy, which is separated in induction chemotherapy, consolidation therapy, reinduction, and maintenance therapy. And we mostly get patients really in complete remission after the induction therapy. So after 5 to 6 weeks, people are losing their tumor, and then you get all this treatment here for the next couple of years. What is also important is we get very early on a relapse. So in 66% of patients, we get an MRD positive case. And this is quite interesting because we acquire also chemo resistance during this time. We don't see any increase in secondary mutations. So the genetics is exactly the same, but we have something which we call immune evasion. There's an immune evasion mechanism, which I show you in a minute, which is quite interesting because the relapse is occurring from a cell which is protected by the immune system. And what is here actually happening is this. We have a leukemia initiating cell, and this is turning on a new ligand, which is well known, but not new for leukemia people, which is ICOS ligand, and which is a checkpoint. And what is happening, this checkpoint is interacting with regulatory T cells, which express ICOS on the cell surface. And this interaction make a reaction because a regulatory T cell is secreting interleukin-10. This causes a rapid expansion of regulatory T cells, which then block the activity of normal T cells. And so any activity of normal T cells to kill this leukemia initiating cell is suppressed by this factor. And interestingly, we came up with this model and this data because we investigated something which we have identified some years ago, namely that T411 infant leukemia, we see a group of patients which are not HOX high, which are HOX low, and they express this homeobos gene Iroquois. And we have found in the last years that Iroquois is turning on a very important transcription factor, which is IGR3, and IGR3 is directly upregulating ICOS ligand. So the story started here with this old paper from 2009, where we identified the Iroquois gene in the HOX low group of patients. And then a year later, the Netherlands group published a story that these Hox-low patients have a higher rate for relapses. This was done with very few patients only. We had 20 here where we did this, and they just confirmed by the clinical data that this is really a worse signature. And then we did a lot of experiments, and what we found out by overexpressing EGR3 in stem cells, where we used EGR3 overexpressing stem cells in mock cells to really show that ICOS ligand is upregulated. And so we had closed the gap. We could finish now the story from which we discovered in 2009. The T411 patients with Hox-low are expressing this factor here, and this turns on this factor, and this turns on this checkpoint. We now analyzed in Germany with the help of people from Germany, from Italy, and from Paris, patient data where we have the outcome. And this is only T411 patients below 1 year of age. So it's really infant leukemia. When we separate the infant leukemia patients by outcome and we look for Hox high and Hox low, we don't see any significant difference between these two groups. But when we separate it according to ICOS low and ICOS high, we get a very strong separation of these patients. So ICOS high means that these people are not surviving. So from this data, we can conclude that high ICOS, this checkpoint expression here at diagnosis already, is really predicting an inferior outcome. We also did another study where we get samples from Berlin, which is only studying relapsed patients. And we had here relapsed patients with different MLL fusions, but they're mostly 80% were AF4. And what is interesting, this is a relapse group. Yeah. So these are really relapsed patients, and we compare this again to the patients from the earlier group where we know the outcome, and you see these are the ICOS low group, and this is the ICOS high group, and the ICOS high group is completely overlapping with the relapse group. And this can be also confirmed by clustering. You see that the red signatures and the green signatures are coming together and are separating from this signature here. So the principal component analysis confirms again that ICOS high expression at diagnosis is relapse predicting, which is also important. And then we did an experiment to validate this omic data. So we isolated T cells from healthy donors and co-cultured them together with our stem cell with MOC or with EGR3. So here's ICOS high, and analyzed what is happening then. So we had a very clear-cut way to analyze regulatory T cells. And we did also cytokine quantification. And we can see that in the cells which are expressing the ICOS ligand, we see an increase in regulatory T cells. So in every patient we were looking at, we see an increase in ICOS ligand expression. When we block by an antibody ICOS ligand, there are antibodies available, then we see an increase and decrease here in regulatory T cells, and we see this also by looking to interleukin-10 production, while interleukin-2 production was not changed. So this means, and this is a paper which came out recently, that ICOS ligand is a new marker which is quite important when you want to investigate leukemia patients. High ICOS ligand expression is predictive for relapses, and this is due to the expression of EGR3. And the problem is it's not only in infant leukemia what we published, other people have published before exactly the same for gastric cancer, melanoma, glioblastoma, breast cancer, multiple myeloma, and AML, that this expansion of regulatory T cells, which can be monitored by interleukin-10 secretion, is predictive for inferior outcome. And so our idea is now, having this picture now in our mind, that if we block somehow the interaction of this checkpoint here, and there is an antibody available, Prevalence, who is disrupting this ICOS ligand-ICOS interaction, should actually be preventive for MRD, because then we should see interleukin 10 secretion, we don't see the expansion of regulatory C cells, and we bring back the normal T cells to their chance to kill the leukemic stem cell in the bone marrow niche. And therefore, we want to do a study now, which is implementing to this chemotherapy strategy the infusion of Prevalumab over a period of time here to see whether we can see reduced or the absence of relapses in the future. So, and this brings me back to my last picture, where I summarize all the efforts on Earth to somehow interfere with small molecules, or now maybe with this antibody, with the progression of the disease. So you can see here, let's say, the famous DOT1L study, the menin inhibition, the Z inhibition, the HDAC study that we have performed in Frankfurt already. Here's a new study which we published last year, that we can block the cleavage of MLL and also of A4ML. And now the idea also is, together with Maribel, to use enhancer of CEST inhibitors also to activate MLL back. And I hope you can understand that this is really a major effort, and everybody of us who is working on the prevention of leukemia should, of course, be interested in finding something which is really enhancing the survival of these patients. So I thank you for listening. And I also want to mention all the funders that we have. And one of the most recent now is the German Josep Carreras Stiftung in Germany, which is funding the new project between Maribel and me. So thank you very much.
Okay, thank you, Rolf. Amazing. From very, very basic, 20 years ago to now to the clinics also. That's nice. So questions.
Thank you very much, Rolf. Great talk. So at the beginning, the first part, this, how you call it here, this early termination, no, the non-genomically encoded fusion transcripts. These are the same that the chimeric transcripts? Is the same, or this is a subtype of chimeric transcripts?
No, this is a functional fusion transcript between ML and A4, which you can see in every blood cell, like BCR-ALO.
Yeah, but when people did RNA-seq, they found a lot of chimeric transcripts between different genes, not only in this subfamily.
Good question, Manuel. Yeah. You can find in RNA-seq experiments, if you look to it, all the gene fusions which are possible may happen, but this is all transpliced RNA based on this co-transcription, these transcription factories. Yeah. And if you have proximity and the ability of transplicing, you can find it. You can find hundreds of these kind of fusions if you look carefully enough in RNA-seq data. Yeah. But they are not encoded by the genome. They are really transpliced fusion transcripts.
And related to this also, so this happened because some of them are in nuclear proximity in interface, you said. But of course, all of us has the same in proximity, but only a few people develop the leukemia, but the proximity is everywhere. Or these people, they are more proximal than others. So why some people develop then the leukemia, others, if the proximity is everywhere in all of us?
This is exactly the statistics. Every day we have a chance to get a leukemia for exactly this reason, but you need a DNA damage situation. You need a DNA damage, the presence of the RNA, and then you get the wrong repair because you're fusing two different chromosomes, because we have also some other data really showing that the RNA is necessary to make a wrong repair.
It's necessary, but you need the DNA damage before to start the process. Yeah. Okay. Thank you very much.
Hi, Rolf, directly following up on this. So you're saying the RNA is really necessary for inducing the wrong repair. So is there a switch between homologous recombination to non-homologous end joining? Is this the next story?
We will publish a paper in the next time really demonstrating this. We have done some other experiments which are really showing that the RNA is a key component for the translocation. So we have, I didn't want to bother you with all this basic science stuff, but we really could clearly show that without RNA, even if you have a DNA damage situation, you don't get any translocation. It's impossible, but you need the fusion RNA to get the chromosomal translocation.
And do we have any understanding what is the evolutionary reason for having early terminating transcripts in our genome in the first place?
That's a very good question. Is there an evolutionary, it could be really just by chance that you have something which is terminating transcription. So it could be by chance, happening in every intron of our genes. This is one possibility. But if you really think about evolution, then you should do a very simple experiment. Compare the MLL protein with Trithorax. Trithorax is the invertebrate homologue of MLL. And I found it very interesting that when you compare the structure of Trithorax with MLL, that it's exactly overlapping with the part which is separated by the translocation, which is the C-terminal part of MLL. It's not the N-terminal part of MLL. So, and what we published already some years ago is there's a promoter driving only the expression of the C-terminal portion of MLL, and not coming from there. So you could imagine that the ML protein or the ML gene during evolution in vertebrates is a fusion of two different genes. The N-terminal part is one gene, the C-terminal part is a Trithorax homologue. And they were fused together in evolution somehow. But there is still the termination coming for the first gene and the promoter for the second part. So it could be also a rational explanation why we see in certain genes this kind of premature termination of transcription, because maybe these genes were during evolution fused. They're actually the product of two genes which are fused during evolution. Could be another explanation for this phenomenon. But it's interesting that all the genes that we have investigated so far, which are known from translocations, have this phenotype. It's really interesting.
Hi, very nice talk. From the last part and the expansion of Tregs dependent on ICOS ligand, I wonder if all the suppression is mediated by IL-10 or there are other mechanisms of suppression. And then as a follow-up, are both CD4 and CD8 anti-tumor T cells affected equally by this Tregs expansion?
What I can tell you from our in vitro data, we have no in vivo data, we see clearly when we co-culture the T cells freshly isolated from patients, we see the expansion of regulatory T cells, and we don't see so much expansion of other T cells. And this is due because IL-10 is a growth factor for regulatory T cells. So there's an autostimulatory mechanism really expanding the regulatory T cells. And there were some other people who have done also some work on this. They clearly show that even in the presence of only CD4 and CD8 positive cells, and without any regulatory T cell, by having this ICOS ligand ICOS interaction, there's a development of new regulatory T cells. So there's a strong push into the direction of regulatory T cells, and we think that in the bone marrow niche, the lick is surrounded by these regulatory T cells and protecting this lick, this leukemia initiating cells, from being destroyed by our immune system. And therefore, if we can interfere with this ICOS ligand ICOS receptor interaction somehow, and we are currently planning some experiments for that to show this, even in a mouse model. In the future, we hope that we can really show that we can destroy the leukemia initiating cell and therefore prevent the relapse. This is our current idea, but we do not know what is really happening to the other T cells. We have no idea.
Hi, here. Thank you for the talk. It's related to the first part. I was wondering if in chromotripsis, that is massive translocations, you also need to have this proximity and this transcription.
That's a good question, because the chromosomes after chromotripsis are repaired somehow. And when I started this project, I had three arguments why I really got into this project. First of all, when we looked to the first RNA-seq data which came out, it was evident that not only genes are transcribed, but also major portions of our chromosomes are also transcribed. So we have non-coding RNA. Our nucleus is full of non-coding RNAs, which has actually no function, but maybe for repair processes. So this was one of my ideas why I was so interested for this project. So we have normal RNA in various amounts, which are copies of genomic DNA. We have in sometimes fusion RNAs when two chromosomes are close to each other and genes have this phenotype of early termination. And all this makes a lot of sense if you think about genetic stability. So if it's true that we need, for example, RNA for DNA repair, nobody has really investigated this so far. If we use copies of our genomic information to repair lesions in our DNA, and this is a normal process, then fusion RNAs are dangerous when you have a double-strand breakpoint and you have a wrong fusion RNA in vicinity, then you get a chromosomal translocation. This was basically the idea behind this project when we started this some years ago. So I think this is really not very well investigated by nobody right now, that RNA is very important for DNA repair. And you can find it in the literature. There are a few people working on this, which is called RNA-mediated DNA repair. If you go in Medline, PubMed, you will find some interesting paper really going into that direction. So maybe RNA per se is a very important source for DNA repair.
Thank you.
Okay, other question? Pablo?
Rolf, very nice speech. Just a quick question. Have you been able to link the final ICOS story and immune evasion with lineage switch to AML in these patients?
We have not treated patients so far, but I think because you mean that we get a downregulation of ICOS ligand when we make a treatment or something like this? Could be. I don't know. We have not treated patients so far, but if this will happen, then the leukemia initiating cell will be highly vulnerable for the immune system. If it's losing the connection to the regulatory T cell in the bone marrow niche, it may not survive, probably. This is the idea behind. So I hope that this is happening. It's really an Achilles heel. You mean this picture? This interaction here seems to be an Achilles heel of the leukemia initiating cell. I don't think that there's a switch coming up.
I'm asking this because, as you may know better than me, we are all blaming the cell of origin as a potential underlying mechanism of lineage switch.
However, yeah, but lineage switches in case of CD19, when you use blinatumomab, the lineage switch is very easy because if you downregulate PAX5 in these cells, which is turned on, if you lose PAX5 expression, you lose CD19.
But there are many lineage switches in patients just treated with conventional chemotherapy.
Yeah, but this is a checkpoint here. It's a different story. It may be really successfully when we are blocking this here that we get really this residual tumor cells eradicated. I hope so. Really, I hope so. We need to test this in the future, of course.
It would be nice to look at. I know Olaf. Olaf has very nice RNA-seq studies.
I know.
In diagnosed last match patients relapsing as ALL or AML. It would be nice to look at this data. Yes.
That's a good point. I can ask Olaf for that. Maybe he can check his databases. What is happening with ICOS ligand here in that case?
Okay, we have some questions from the virtual audience. The first is a terrific talk, thanks. First question, have you observed this cooperative transcriptional effect of these fusion proteins in other contexts, like for MLL AF9 leukemias?
I assume that MLAF9 is completely different. In MLAF9 and also MLE, they really work as a standalone protein. So this is quite clear, but they are causing, if they are working as a standalone protein, they are causing always acute myeloid leukemia. It may be interesting to investigate this for MLL, which is causing in patients acute lymphoblastic leukemia, but in any mouse model, acute myeloid leukemia, when you're using it alone. So it could be that the same will happen when we use both together, that we get also ALL instead of AML. So that the cooperation between the fusion proteins is causing a distinct disease phenotype.
And are these transcriptional changes maintained after the 48 hours of induction, or they are transient, like in the AF6 example?
We have investigated everything after 2 days, instead of this 4-week study, what I showed in the beginning. It's maintained, when you look after longer time points, it's maintained, it's always changing. So you see the evolution is going on, but I cannot tell this for the T611 because T611 we have investigated only for 2 days of induction. So I cannot tell you what is happening after 4 weeks, for example.
Okay, another one is, since we know that for AML and ALL age of patients is very important, how do you connect this with the data that you showed about the breakpoints and their capacity to switch from AML to ALL?
Oh.
Long question.
So, yeah, but I have to mention that the fusion proteins we are using are exactly that one of infant ALL. So the breakpoints are exactly from those of infant ALL patients. We have no breakpoints used for adult patients because the breakpoints of adult patients are mostly in ML intron 9, while we are using only intron 11 fusions in our lab. So I cannot really answer this question because we never have investigated ML fusions with intron 9, except for the T611 situation where we have only the fusion in intron 9 to get AML. And if you use the other breakpoint, we get TALL, presumably. So the breakpoint then defines disease phenotype, and it's also maybe linked to the age of patients or person.
Okay, so that's, there is another, just the last question, but that answer leads me to a conceptual question that I have. So we both work with T411 infant, Pro-B ALL. So conceptually, what is the difference with the adult? So the infants are doing there is a very dismal prognosis. The conceptual from angle, what happened with adults with this exactly the same T411?
Yeah, but the origin, the cell of origin is different. The cell of origin in infant leukemia is a fetal liver cell, and the origin of an adult leukemia is presumably a bone marrow cell. And there could be quite big differences in the biology.
Okay, and the last one is, are MLL, I think you showed that already, MLL rearranged able to generate secondary mutational events?
People are describing secondary mutations, but they are not necessary. MLL rearranged leukemias are really, they have the fusion proteins and they don't need secondary mutations. Not only we, also others have published, for example, that sometimes you find RAS mutations, but it's funny that you see a RAS mutation at diagnosis and then relapse material, it's gone. So it doesn't exist anymore. So you have subclones with submutations, but they are not really important. We need only the fusion proteins.
Okay, so thank you very much, Rolf, and thank you all for coming to the talk.